Hi, my name is Justine Whipple, and I'll be presenting on Leigh syndrome this evening. The date is 10/23/24. Leigh syndrome is a neurometabolic disorder that attacks the mitochondria in the body. Today, we're going to go over the impacts of the disease, the prevalence and the inheritance of this disease, the diagnosis and how this syndrome is determined, the therapy and treatments that are currently available, and the future of what might be possible in treating this syndrome. Of note, the quote on the bottom talks about the fact that this is the most frequent pediatric clinical presentation of mitochondrial disease, meaning that there's many different types of mitochondrial diseases, but this is the one that seems to present itself the most. Additionally, there is Leigh syndrome, and there is also Leigh-like symptoms. So there are many symptoms that mimic some of the issues that are associated with the syndrome, full-blown syndrome. There is still a lot to be learned about this disease. First, we'll start discussing the impacts of the syndrome. What is it, and how does it affect our body and the systems within? As a neurometabolic disorder, this disease attacks our metabolic pathways in three different primary manners. The first way is that it degrades ATP production that's critical for our cellular production and development by disrupting our Krebs cycle. Second of all, it interrupts our electron transport chain, which is so critical in complex 5 for producing all that ATP that's necessary in the oxidative phosphorylation. And finally, it can impair the pyruvate metabolism, which once our body creates lactic acid, it's unable to process it and turn it back into energy. All three of these areas can decrease the energy levels that are critical for cell development and normal development of the body. While there are many different aspects of your body that this impacts, the primary ones are your brain, because this is where it presents first. Our brain is super sensitive tissue, same with our spinal cord and our kidneys. Degeneration and brain cell lesions start appearing in small children. There's muscular hypertrophy and muscles that don't develop correctly, as well as increased kidney function is another symptom that patients often present. Finally, spinal cord and optic nerve degeneration are also highlight signs when in conjunction with those other symptoms that patients may present. This is a great overview of the presentations of Leigh syndrome. As you can see, it impacts every system in the body because ATP is so critical for all of the development, regeneration of cells, and normal daily living within a human body. On here, you'll see that normally it is early onset, which affects 1 in 40,000 births, which are generally presents in under 2 years old. In very few cases, it may present in older individuals and may actually progress slower than in some of the pediatric patients that incur Leigh syndrome. So, impacts of the disease. The impacts on our bodily functions is that it degrades aerobic respiration, meaning that our muscles are going to be weak, we're going to have diminished tone, hypotonia, which then can lead to all of these other issues with degraded muscle coordination and activation. So a number of different symptoms, including dystonia, which is disconjuncted, potentially muscle jerking, twitching, ataxia, tremors. All of these things can be associated with Leigh syndrome because of the lack of development and the degradation of our bodies. Muscle weakness and diminished muscle tone can directly contribute to some of these other symptoms, as well as degrading our vision. So nystagmus and vision loss are often associated with the diagnosis of Leigh disease. In terms of cell cycle failure or impact in regarding to lifestyle, unfortunately, our lifestyles are really not determinant of this disease, nor is the cell cycle. So there isn't any way right now that scientists have been able to get in and manipulate cell cycles to prevent or reduce the impacts of this disease. Right now, we know that Leigh syndrome is caused by the alternation of protein complexes that are involved in the oxidative phosphorylation, which is caused by the variants in genes that direct the replication or production of mitochondrial proteins. So these are essential proteins that allow oxphos to occur. An example of this is the complex 5 in the ETC, which is critical for generating the most amount of ATP. In terms of lifestyle, as mentioned before, unfortunately, there's nothing that we are able to determine that is causing this disease in individuals, small children, or adults that could be altered to lessen the impacts, or lessen the chance of individuals acquiring this disease. The diagnosis. How do we diagnose this disease? We diagnose this disease through magnetic resonance imaging, so MRIs or computed tomography, which are taking small slices and scanning brains. As seen in the original photo, scientists and doctors are able to determine where there is degeneration in the brain and identify those lesions that may appear, as well as cysts that can present in the cerebral cortex. This can attack the brain in a number of different ways or present in the basal ganglia, the brainstem, or gray matter, which is obviously critical to the development of a child. We can also identify it through a number of laboratory tests in conjunction with these MRIs and CTs. So if we find acidic waste products as an indicator, elevated levels of pyruvate, as earlier discussed, because we're unable to process it, and alanine, low blood sugar, and metabolic enzyme activity can all be indicators for Leigh syndrome. Finally, a conjunction of doing a biopsy of the muscle, as discussed with the hypertrophy of muscles, and genetic testing can bring doctors to a conclusion that a patient is affected by Leigh syndrome. What causes the syndrome? It's caused by mutated genes, which originate in either the nuclear genome or mitochondria, or there's a nuclear mutation. So there's two different variants that can cause this disease. With nuclear genome, we're talking about the nuclear proteins. In terms of the mitochondrial proteins, both of them are critical to our ATP synthesis and development of muscles and bodily tissues. And here you'll note that these mutations, the 8993T and 899TC, are both mutations in the MTATP6 gene, which encodes subunit of the ATP synthase. As earlier mentioned, this disrupts oxidative phosphorylation. There's 25 genes part of the electron transport chain, which may be associated with Leigh syndrome, making it really challenging to diagnose and to treat. Most often, though, doctors have seen that the gene that's altered in Leigh syndrome is SIRF1, which is surfeit locus protein 1. It's located on chromosome 9, and it's important for providing the instruction for proteins critical for oxphos and aiding in the correct assembly of the complex 4 protein enzyme that is in the electron transport chain that enables ATP to be developed. The implications of these mutations is that smaller than normal proteins are produced, which then are broken down by the cell and the enzymes faster, which causes a loss of function with the SIRF1 protein, which directly impacts the ETC and the oxphos cycle. Without this protein, the COX complex, which is the fourth complex within the ETC, is not able to perform its function to produce ATP. The mitochondrial ATP gene provides instructions for building a portion of the complex 5 in oxphos. So variants of the mutated MTAT6 gene can block the generation of ATP completely or decrease the activity of the oxphos complex, impairing energy production. As mentioned before, the mutation implications are that brain, muscle, heart, and cells are all particularly sensitive to a decrease in cell energy and will be impacted by development and sustainment. Here's a great overview of the ETC and where it causes issues, where Leigh syndrome causes issues. So I'll draw your attention to here, where we have cytochrome C oxidase, which is unable to be produced with a dysfunctional protein, and the inability to direct on to complex 5, or the disruption of the building of this complex, which results in our full ATP cycle. Right now, there's molecular genetic testing that's available, which allows for sequencing of mitochondrial DNA or nuclear DNA. This allows scientists and doctors to look at and determine what is going on with this syndrome and how they might be able to get at adjusting these proteins and combat and come up with a cure. Right now, there's also there's targeted single gene testing. So you can either do a full genomic testing, which is super expensive, or targeted single gene testing, which looks at common pathogenic variants. Unfortunately, because there's so much variety, it's really hard to narrow it down to the single gene that's causing these issues. And it's very much dependent on age, availability, and genomic testing. All right. The prevalence inheritance. This disease is passed on via heterogeneous heritable disease, which means that it is passed on via autosomal recessive pattern via nuclear DNA, which means both copies of the genes must have to have the variant for the phenotype to present. This is applicable to most genes associated with the Leigh syndrome, including that SIRF1, and this is the primary way that patients inherit this disease. About 20% of the population that's affected that have Leigh syndrome have inherited it via mitochondrial pattern, which means that only the mom has the trait, and the egg is the one that contributes to mitochondrial development. So that means that both the mother and the son would be affected because of that X gene being passed along. So the three different modes are autosomal recessive, mitochondrial inheritance, or X-linked recessive inheritance, which means that it'll be only inheritable by the males and the offspring. What can we do about this? Right now, there's no cure or specific treatment where doctors are primarily focused on treating symptoms versus curing. The targets for therapy are reducing oxidative stress, restoring oxidative phosphorylation, and then treating other symptoms include the degeneration of vision, breathing, epilepsy, and tremors. So the most effective means of treating the symptoms have become in the form of either diet or supplements. Ketogenic diets that promote fatty acid oxidation, which is an alternative for oxidative phosphorylation for energy, has been used. A number of supplemental cocktails, so combination therapy has been found to be more effective than just individual supplements. Adding sodium bicarbonate to increase the efficacy of the energy cycle, and then supplements such as CoQ10, riboflavin, L-carnitine are all critical, and so essentially supplementing the shortfalls of where ATP and aiding along trying to assist the ATP production within the cells. Here's a great overview of the targeted mechanisms. You can see where they're injecting, where the biotin is critical here during the electron transport chain before it reaches complex 1. The insert of the Epi743, which we'll talk about here in a moment. How the ketogenic diet comes into play in terms of providing and going to the citric acid cycle, and here within the alternative bypassing of the electron transport chain. How can we help? So the best ways to help our patients right now is through education, advocacy, and supportive measures. Taking care of the people that we love, providing them with these supplements, which is shown here, showing thiamine was effective, biotin, it's just showing, and the ketogenic diet. These were all not great for assisting, but they were assistive in terms of helping out our patients. And the future, what does the future look like? There's a symposium that meets to discuss how to get around solving and coming up with a cure for this disease. Recent gene editing technology with a mitochondrial model has been looked at. Current clinical trials have been tried on, but really the focus is on mitochondrial health and ATP production to kind of get around the tissues, because the issue is our tissues that are degrading. So our clinical trials that are going on right now are some that are highly controversial, where they're replacing oocytes that have mutations with donated oocytes. and then as well as the different clinical trials that they have tried for therapy and augmenting mitochondria and just supplementing different aspects of this cycle. That concludes my presentation today regarding Leigh syndrome.